Transcription, the basic concepts of transcription. We will look at the template and non-template strands. Remember, when you go to the replication, the two strands of DNA will act as the template at the same time in almost all the genomic DNAs. Only in the case of some viruses we see the difference where one strand acts as a template and at a later stage second strand acts as the template. In replication, both the strands have to be replicated at the same time. In transcription, for any given transcription, one strand will act as a template strand, another strand will act as the non-template strand. Now, how you identify which of your DNA strand acts as a template and which of your DNA strand will be acting as a non-template? This depends on a regulatory sequence which is called promoter. A promoter is a cis-regulatory sequence. Cis means it will affect adjacent region. Cis-regulatory sequence, that's one function it is. It will be affecting the adjacent region and is the binding site for RNA polymerase during transcription initiation. So it is doing two things. One, it is acting as a regulatory unit. It is controlling the transcription of a downstream region. The second side, it allows the binding of the RNA polymerase at the promoter site so that you can have a stable binding of the RNA polymerase and therefore you can have the transcription process successfully initiated. That will be done by RNA polymerase holoenzyme. So it is the direction of a strand with respect to the promoter that determines template. So the rule is the orientation of a given DNA strand with respect to the promoter sequence decides whether this strand acts as a template strand or it acts as a non-template strand. So let us say we have an arbitrary sequence which can act as a promoter. So that is the promoter sequence. I'm not drawing any sequence here, but I'm indicating that this is the promoter and this is the place where RNA polymerase binds at the time of initiation. Then we have the two strands of DNA. So let us take this strand. We'll put some sequences A, T, A, G, C, A, C, T, A, so on. So I have just taken 3 codons to explain the two strands. This strand should have a complementary sequence T, A, T, C, G, T, G, A, T. So now we have drawn the two strands from the promoter. We have made the complementary strands, complementary nucleotides. Let us say this strand is 5' to 3'. This strand is 5' to 3' in the opposite direction. So let's carry out the transcription. So we have to decide which one is template. The strand which is oriented 3' to 5' with respect to promoter will be the template strand. This is the simplest law that you have to remember. The DNA strand which is oriented 3' to 5' with respect to the promoter will be acting as a template strand for a given transcription. Whereas the strand which is oriented 5' to 3' with respect to promoter will act as non-template strand. So that's a conventional way of defining template strand and non-template strand. So the DNA strand which is oriented 3' to 5' with respect to the promoter is the template strand. The DNA strand which is oriented 5' to 3' with respect to the promoter is the non-template strand. So when I do the transcription, I get an RNA. So RNA starts with 5', gets complementary nucleotides which are ribonucleotide. So here we get A, here we get U, here we get A, here we get G, here we get C, here we get A, here we get C, here we get U, here we get A, 3'. This is your mRNA strand. So this is template. This is non-template. Now look at the two strands, the template strand and the newly synthesized RNA strand. Template strand is oriented 3' to 5', whereas mRNA strand is oriented 5' to 3'. That means their orientations are opposite. Their orientations are reverse. That's why template strand is called as anti-sense DNA strand. Its sense is opposite to that of the RNA. Next one, look at the nucleotides. Template strand has TAT. Here you have AUA, you have CGT, you have GCA, you have GAT, you have CUA. What it means is the template strand has complementary nucleotides compared to the RNA that is synthesized. It is not having identical nucleotides. That's why this strand is called non-coding DNA strand. So now we got the three different names for one strand. Template strand, it is oriented 3' to 5' with respect to the promoter. It provides the complementary nucleotide sequence for the synthesis of RNA. It is antisense DNA strand because its orientation is opposite to the orientation of the RNA transcript. It is non-coding DNA strand because it has a nucleotide sequence which is non-identical but complementary. Now what happens to your non-template strand? Non-template strand is 5' to 3'. RNA strand is 5' to 3'. So the two have same orientation. So we call this as sense DNA strand. They are sense DNA strand because orientation is similar to that of RNA strand which is synthesized. Now look at the nucleotides. We have ATA, AUA, GCA, GCA, CTA, CUA. The nucleotide sequences are identical. The only difference being DNA contains thymine in place of uracil in the case of RNA. That's why this strand is called as coding DNA strand. So again, it's called as coding DNA strand because its nucleotide sequence is identical to the RNA sequence except that T's are replaced by D. It is called sense DNA strand because its orientation is exactly same to that of the RNA strand. The next thing that we can do here is we can identify codons. So you have here the codons. These we call it as DNA codons. So ATA is DNA codon, DNA codon, DNA codon. Whereas these we call it as mRNA codons. So codons we can find in non-template strand of DNA as well as the mRNA. Then what about this in the template strand? These we call it as anticodons. So anticodons we can find in template strand of DNA as well as in the tRNA. So that is one anticodon. This is another anticodon. And this is another. These are all tRNAs. And if you see that the tRNA anticodon is exactly similar to the anticodon that is present in the template strand of DNA, except that RNA contains uracil, DNA contains thymine. So TAT, UAU, CGT, CGU, GAT, GAU. Now when you go for pairing anticodons, you write from 3' to 5'. For example, if your codon is AUG, that codes for methionine. You write 5 prime to 3 prime. Your anticodon will be should be complementary, so it comes 3 prime. A for U, U for A, G for C, 5 prime. But we get a problem here, and that problem is any nucleotide sequence should be written from 5 prime to 3 prime end. So writing this anticodon, what we will do is this. We write 5 prime C, A, U, 3 prime. And then we show a reverse arrow. Wherever you are seeing that reverse arrow, it indicates an anticodon. So this is anticodon of methionine codon. This is methionine codon. This is methionine anticodon. So those are some conventions that we have. So we clarify the concept by taking a problem. A part of mRNA sequence. So I am not writing the total, but I am just putting a small portion of that. Is 5 prime AUG CGG CAG GUC 3 prime. Write first one, template strand sequence. Second one, coding strand sequence. So first you can note down the sequence which is given and write it as RNA sequence to avoid any confusion. So this is the RNA transcript. Now we can write the coding strand by directly copying down that and replacing U's with T's. So 5 prime, A will be A, U will be T, G will be G, C will be C, G will be G, G will be G, C will be C, A will be A, G will be G, G will be G, U will be T, C will be C, 3 prime. So that we got coding DNA strand. Now we have to go for the template strand. The template strand will be with respect to the promoter 3 prime to 5 prime. So 3 prime A we get T, U you get A, G we get C, C we get G, G we get C, G we get C, C we get G, A we get T, G we get C, G we get C, U we get A, C we get G, 3. So this is the template strand. Now even actually you can show it with promoter. So promoter, so one end of the promoter you write 5 prime, second end you write 3 prime. First write coding strand A, T, G, C, G, G, C, A, G, G, T, C, 3 prime. Now second side write template strand. It will be automatically 3 prime. T, A, C, G, C, C, G, T, C, C, A, G, 5 prime. So promoter, template, non-template. Now one thing that you have to remember in the exam is when they ask you to write a template strand, they will ask you to write in 5 prime to 3 prime direction. So this we will write. This is important. They won't give you 3 prime to 5 prime. They ask you to write in 5 prime to 3 prime. So just we'll go back to this side. We write 5 prime, then GAC. You can take in terms of codons. CTG, CCG, CAT. It's over. You got your template strand. So always when you write the strands, follow 5 prime to 3 prime directions. Especially when they are asked to find out any one particular strand, you are expected to go from 5 prime to 3 prime direction, because the convention in molecular biology says we should write sequences always from 5 prime to 3 prime direction. That is the direction in which the nucleic acids are